Okay, we're going to start. So thanks everyone for joining. I think we'll expect to have a few more people coming, but it's a real pleasure to introduce Professor Chen today. Professor Chen is one of the leading figures in studying epidemiology of Parkinson's disease. He started quite a few years ago, originally working with Alberto Esquerro at the Harvard School of Public Health, where he did some of, I think, the most durable work in the field, looking at modifiable risk factors. He subsequently went to the National Institute of Environmental Health Science, where he was an investigator there for a number of years, and then joined the faculty at Michigan State, where he's continued to work on Parkinson's disease. And he's going to tell us today about his continuing research on environmental risk factors for Parkinson's disease, which is a really interesting and fairly dynamic field. So thanks very much.
Thank you, Roger, for the kind introduction. And I probably will say here too. Yeah, okay, yeah. And it's really a dynamic field of research. And you probably can see that from my presentation, how my interest in studying Parkinson's disease epidemiology changed from one stage to the next. So today, I hope I can share with you my thought about the environmental epidemiology of Parkinson's and presenting you some of the challenges and potentially strategies to better understand the etiology of Parkinson's disease. And oh, yeah, I probably need to do this. Yeah, okay, it works. Okay, thank you. Yeah. So, you know, for those of you who pay attention to Parkinson's epidemiology, probably this is, you know, not something new, but for others, I just want to give a little bit background so we can be all on the same page. You know, this is the data published a couple of years ago by Dr. Reed Dorsey. Sort of giving the estimate of Parkinson's disease patient, we're talking about the number of patients or the prevalence of Parkinson's disease. Back from 1990s until like 2015, 2016, the estimated number almost doubled. And then projected into the future is probably will be double in another 25 years. So the data, you know, suggesting that the burden of Parkinson's disease increased in the past 20, 30 years, and probably will continue to do so in the future, representing increasing burden of the disease in the population. And, you know, if all this is true, it probably cannot be explained by age alone, because there are some evidence, although less consistent, suggesting there is a probable increase in the incidence of Parkinson's disease as well. And if this is true, it may not be explained by genetics, because genetic may not change within such a short time period. Probably environmental factors may play a role, probably gene-environment interactions, and probably genetic epigenetics regulations of the genetic expression. So, you know, put the genetic aside, how about the environmental risk factors? And the answer is we do not know much about it, and mainly because of the inadequate research on environmental risk factors for Parkinson's disease. So this the data, again, published by Dorsey and colleagues a couple of years ago, and looking at the publications from the 1960s, the past 60 years, essentially, you can see compared to the genetic research, the environmental research has been very few compared to the genetic research. And also the genetics has been making a great breakthroughs in understanding the genetic causes of Parkinson's disease, but the environmental understanding has been lagged behind. So we do not know a lot, but environmental risk factors really important for Parkinson's disease. Most of you probably will be agreeable to this idea that environmental risk factors is important. And about 25 years ago, this seminal paper was published, conducted by Dr. Kelly Tanner, and looking at the concordance rate between monozygotic twins and dizygotic twins in terms of Parkinson's diagnosis. And what they found out is among the early onset cases, the concordance rate in the monozygotic twins is much higher than that of the dizygotic. But if you look at cases diagnosed after age 50, or the so-called late onset sporadic Parkinson's, the concordance rate was fairly comparable between the two, suggesting that genetic factors play important roles in early onset Parkinson's disease, but not likely in late onset, or only a portion of the late onset Parkinson's could be attributable to the genetic causes. And the study was conducted in a fairly large twin registry of the World War II veterans cohort. And so 25 years later, the cohort almost had a complete follow-up, and Sam Goldman and colleagues reanalyzed the data with complete follow-up of the study participant, and the findings initially unchanged. So after this publication, there are a lot of interest to understand the environmental epidemiology or the environmental contributors to Parkinson's disease. 25 years later, what did we learn? We had a lot of findings. In epidemiology, if you look at, we have a lot of factors potentially associated with a lower risk Parkinson's disease identified. The most notable one is cigarette smoking, that smokers had a 50% lower risk of Parkinson's disease compared to never smokers. And this finding was almost identified in every single epidemiologist published epidemiological studies of Parkinson's disease. To a less extent, coffee drinking, physical exercise, urate, the use of NSAID, and other medications have also been linked to a lower risk of Parkinson's disease. And also there are some findings on inverse associations between blood total cholesterol and some other dietary components that may be related to lower risk of Parkinson's disease. If you look at the site that associated with higher risk of Parkinson's disease, pesticide probably stands out as the most consistent findings for Parkinson's disease. And some other factors also identified. The dairy consumption of milk drinking has been linked to a higher risk of Parkinson's disease. The organic solvent TCE, head injury, inflammation, virus infections, heavy metals, and more recently, air pollution have all been linked to higher risk of Parkinson's disease. So if you look at the number of findings and the kind of consistency of the findings, is fairly remarkable. There are a lot of epidemiological findings in relation to Parkinson's disease risk. So a lot of findings, but are there any benefits? The answer is quite disappointing. Probably not much benefit we have gained from the studies of Parkinson's etiology. So what's the problem? As epidemiologists, we know that in population-based studies, we are studying associations. Associations does not imply causation by itself. And there are a lot of difficulties in the in establishing a causal relationship. And this probably more evident in Parkinson's disease studies. The epidemiological findings are likely to be caused by a lot of biases, like confounding selection bias, attrition, recall bias, and reverse causation. I'm going to touch upon this. And also the environmental exposures are a lot more difficult to measure as compared to genetics. So you got a very long time window to study Parkinson's environmental exposures and their mixtures. And all these issues could be compounded in the Parkinson's disease epidemiology research due to the lack of understanding about the decades of the so-called Parkinson's disease prodromal development, which is this. So we all know that Parkinson's majority of the cases are late onset, let's say age 80, but the disease does not start like age 79. There might be decades before the disease diagnosis, during which many environmental factors may come into play, either to initiate the pathology or to modify the disease progression process. However, in the past, until like 20 years ago, this has been a black box to many of us. And the lack of understanding about the Parkinson's prodromal development, the 20 or 30 years where the disease pathogenesis develops and progresses, and the lack of understanding of the process by the scientist has made it really difficult to study Parkinson's environmental etiology. And we can use smoking as an example to show you how difficult to understand to establish a causality. I mentioned earlier that smoker had about 50% lower risk for Parkinson's disease, and this is a well established association. However, how to explain the association has been debated since the beginning. It is a causal relationship that smoking or the behavior of smoking actually decrease the risk of Parkinson's disease. Or actually, this is a reverse causality that people who are on their way to develop Parkinson's disease in the prodromal stage, they simply quit smoking. Or it can go back to time that they probably do not start smoking in their early life, or it can be a confounding by their personality or some other reasons. So there are equally plausible competitive hypotheses as compared to the causal possibility. I'm giving the example for smoking, but the same analogy can be made for many of the lifestyle factors: coffee drinking, exercise, alcohol drinking. We publish on everything, but we are unsure about everything. So how about, oh, sorry, I probably need to get back. This may even true for biomarkers. If you still remember what I presented a few minutes ago, that a higher blood cholesterol was associated with a lower risk of Parkinson's disease. But what does this association mean? Excuse me a second. So this is a study we conducted in this is analysis we conducted using the data from the Health ABC study, where they follow up the participants, general population participants, with annual clinical visit and sometimes biannual clinical visit for a long time. And every time they measure the blood cholesterol, and we identify the Parkinson's cases, and we realign the time point in reference to the time point of PD diagnosis and try to see in the Parkinson's patients how their cholesterol levels change in reference to the controls. This reference line is control. The solid line is for the Parkinson's cases. This is modeled. We can see that starting about like 10 years before the diagnosis, you already see a lower cholesterol level in PD patients. These are not PD patients yet. This is well before the diagnosis of Parkinson's. But you can see among this small group of people, their cholesterol level begin to decrease, persistent into the diagnosis of Parkinson's disease. So this is cholesterol. And the other thing is we found is body composition. In this case, the body fat mass measured by DSXA. The same study population, the same analytic strategy, what we found out is not linear. It's like the body fat composition of Parkinson's patients was not differential from that of the controls, fairly stable until the time of PD diagnosis. And after that, it's on a journey of persistent decrease in their body weight, as well as their body fat mass. By now, hopefully, I can present you the challenges of study Parkinson's disease, mainly related to our understanding, our lack of understanding of the decades-long prodromal stage of the disease development. Again, many factors can come into play, either alone or interactionally, synergistically. And many of the things can happen during this prodromal stage, including the biomarkers as well as some of the prodromal symptoms, which I'm going to talk about. And they may affect the exposure assessment and also the analysis of the associations. A related question in Parkinson's disease epidemiology is, besides the study design, how do you measure the exposures? What are the environmental factors you want to measure? There are going to be tons of them during the 20 years of life. And when and how to measure those exposures? Are there any time window that confer more risk than others? So these are all the standing questions we do not have an answer for yet. It's pretty, I present a lot of challenges, and what are the hopes? So it's going to be really tough, but it's not hopeless. And I'm going to present to you one potential strategy that I'm kind of invested a lot of my research interest into it right now to study the prodromal symptoms of Parkinson's disease. This is a paper published by Ron Postumus group that the black box of Parkinson's prodromal stage, we used to think insidious disease onset. But it's probably not, we learned in the past 20 years, it's not a insidious, and it's not probably not a black box anymore. We begin to understand what happened during those 20 years. A group of non-specific symptoms, non-motor symptoms can arise during those time period at different time point. This is the estimate from one study, but you can see that about 20 years ago, the hyposmia becomes to develop in the Parkinson's disease patients, in many Parkinson's disease patients. The REM sleep behavior disorder, the erectile dysfunction in men, constipation, daytime sleepiness, anxiety, depression, they can all occur in the early stage. Before the clinical diagnosis, we call that prodromal symptoms of Parkinson's disease. And the time zero right here is about the time of diagnosis. We can see this non-motor symptoms probably develops decades before the motor symptoms become evident in Parkinson's natural history. So is there any possibility we can put everything together to better understand Parkinson's etiology? So my thought at this time is, can we put the non-motor symptoms and probably the subtle motor symptoms into the black box of Parkinson's disease natural history in the prodromal development in a sequential manner, and try to bring in higher resolution to the black box and try to use them as an intermediate phenotype to understand the environmental etiology of Parkinson's disease. And as far as we consider about the environment, and this may give another a great gateway to study the environmental causes of Parkinson's, because putrefaction is probably the earliest and the most important, most prevalent prodromal symptoms of Parkinson's disease. And constipation and GI disturbances are the others. So we know that the environment kind of interact with our body in the nose and also the gut. So this may help us to better understand Parkinson's environmental etiology, not only what, but also how the environmental contaminants get to the body and then contribute to Parkinson's disease etiology. And along this way, there are a lot of good novel hypotheses being proposed, like the gut to the brain hypothesis in Parkinson's, and also the gut microbiome and Parkinson's etiology. So there are a lot of new ideas can come out from this kind of paradigm to study Parkinson's disease. So about 10 years ago, I began to think about this really carefully. Not carefully, I learned bit by bit to how to develop it, studies to better understand it. But starting about 10 years ago, I'm trying to put everything together and try to develop a staged and cohesive approach to study Parkinson's disease etiology. And essentially, this is another way to present you the ideas I just presented a couple minutes ago. So we have the ideas, but how to do it? And are these ideas really doable and feasible? Because, we need to follow people for like 20, 30 years. I think not necessarily we need to build our own cohort to study Parkinson's, simply because Parkinson's disease is rare. So what I'm thinking about is, what I was thinking about at the time is to leverage the resources of the so-called large environmental cohort. And I'm going to show you two examples of those cohorts. Theoretically, there are a lot of advantages of doing this, because somebody else already collected decades of environmental data as well as health data, and they were collected prospectively. It's already 10, 20 years, so it saves you 10, 20 years of time. And often those studies started, a lot of the participant is still in their midlife. So you have midlife to late adulthood exposure levels already collected. And at the time of our study, probably a lot of the participants already either at risk by age or exposure for Parkinson's disease. They probably already 65, 70, they begin to, the clinical aspect of Parkinson's probably begin to be evident in a lot of this population. And also, a lot of the environmental cohort, they love add-on studies, and they have a strong infrastructure support for add-on studies. But there are also challenges. The challenges being that these studies not designed to study Parkinson's. So a lot of the exposures you might be interested. I'm interested in pesticide, but pesticide not available in many of the cohort. And we are interested in air pollution, but the air pollution exposure may not be the best estimated in those cohort, or the study population may be in just a geographically limited area. So there are a lot of limitations of the study simply because the study is not designed to study Parkinson's. And also for the same reason, they do not have a very scientifically designed strategy to identify Parkinson's disease. And also a lot of the clinical exams for Parkinson's diagnosis is infeasible to do in this cohort. Finally, even though you have a large population already at their age 65, 70, it still take about like 10 years to have sufficient number of Parkinson's cases to study. And NIH does not like this, typically do not have the funding cycle to support a very long follow-up. And in a large population-based study, attrition and non-study participation are always concerns. So I was thinking about, how to overcome some of these challenges. We have to face the reality. Probably we do not have enough financial resources to follow up the entire population. We cannot do everything we want to do. So what I'm thinking about is, can we identify sub-cohort within the big environmental cohort and identify the so-called high-risk population? And we then can focus our data collection for PD research in those populations. And still we take advantage of the exposures they already collected in the parent cohort. But we can do prospective data collections for the exposure to collect biospecimen blood or fecal samples. And we can do more screening for motor and non-motor symptoms, maybe biannually or even annually. We can use some novel clinical assessment tools, telemedicine. I'm going to show you example, some remote device, and we potentially can engage participants to minimize attrition. So it's it's tough, but it's hopeful. I'm going to show you two examples of environmental cohort that I'm collecting relevant data in those cohort. So one is the NIEHS sister study. For those who are not familiar with this study, it's a nationwide cohort of it's a nationwide cohort of women that designed specifically to study the environmental risk factors for breast cancer and also other chronic diseases. And the study started in 2003 to 2009 and recruited about 50,000 women with very detailed environmental data and sample. I'm going to show you in a minute. And the study participants are from all over the United States, all 50 states, as well as Hawaii and Alaska. And after that, every 2 to 3 years, the first follow-up survey was 2 years after, and then 3 years after. And they conducted the first follow-up 2 years later. Not much, things specific to Parkinson's. Starting from a follow-up follow-up 2, which is 2010, 2013, that's the time I began to interested in the non-motor symptoms of Parkinson's. I was fortunate enough to add in motor and non-motor symptoms into this cohort. And then again, in the follow-up 3, and they just finished the follow-up 4 survey of the study participants. And this is the kind of environmental data they already collected. Very extensive environmental data collection. And the air pollution model was just like fresh from the shelf with the most sophisticated air pollution modeling. And they also have the cohort-wide biospecimen data collection from pretty much all 50,000 people. Their blood, DNA, some extracted, others not, house dust, urine, toenail, and diet and lifestyle. And they are performing linkage to toxic release database. And they have extensive and detailed health data. This is mainly by a questionnaire and annual health update. And I began to adjudicate the Parkinson's cases a couple of years ago. We just finished the first round of adjudication and began to do, additional adjudication. And from the second follow-up, we begin to collect these symptoms. So I joined MSU in 2016, and we were fortunate enough to have a grant in 2017 to look to identify a subcohort for Parkinson's research. So in 2018 to 2019, we tested the sense of smell. Remember, sense of smell is the most important prodromal symptom of Parkinson's disease. We tested the sense of smell of 3,400 women, and we also conducted GWAS assays of their genetics. So this is a kind of data collection. We have the entire cohort data. We also have this sub-cohort data, but only at one time point at this time. So I'm going to show you some the so-called non-motor symptoms in relation to Parkinson's disease diagnosis in the entire cohort. These are the self-reported symptoms because we have cohort-wide data on the self-reports. And some of the symptoms, poor affection and dream enacting behavior and constipation, daytime sleepiness, is kind of, we added specifically to study Parkinson's. All these symptoms were included in the cohort for the general research purpose, not specifically for Parkinson's. And in addition to those on the table, there are also a list of other symptoms. I take a look at it, joint pain, short of breath, that kind of questions. Based on the literature review, I think they are not related to Parkinson's. I may be wrong. So I put them as a negative control symptom. And we are looking at self-reported symptom at the follow-up 2 in relation to the Parkinson's diagnosis after follow-up 2. So incident cases, baseline as symptom. Not surprise, almost all these symptoms we know that probably occur commonly in Parkinson's patients is associated with higher risk of the disease. Looking forward, incident. And these are well known. Poor affection, dream enacting behavior, constipation, daytime sleepiness, depression, anxiety. There are plenty of publications, but not so much about fatigue, about weight loss, about dry eyes, dry mouth, about the pain. It's not a hugely strong association, but they show pretty interesting association. And importantly, only one of the 8, we call it negative control symptoms related to Parkinson's, suggesting the report of these symptoms is kind of reliable and sort of specific. And we also add 4 motor symptoms in the questionnaire, and the odds ratio is 1.6 to 5.9, sort of comparable to some of the non-motor symptoms, which means, it's probably fairly strong associations with some of the non-motor symptoms. So these are the symptomatic data, and I'm going to show you some data, mainly just to demonstrate you the data quality and some preliminary findings and how we can study environmental epidemiology in this cohort. They are not necessarily one slide related to another. This is air pollution and incident Parkinson's, again in our cohort-wide analysis. The literature is fairly limited and mixed, and some identify associations with PM2.5. More recent studies identify associations with NO2. We analyze the data in multiple ways in our cohort, and we identify a fairly robust association with NO2, nitrogen dioxide, but not with PM2.5. In the analysis for PM2.5, we do have some findings in subgroups, but I'm not going to get into the details of it. So this is air pollution and incident Parkinson's disease. There are some signals. How about olfaction? Because, it was my hope that we identify an association between air pollution and Parkinson's, and then we identify association between air pollution and olfaction. And bingo, we connect everything together. But it's not the case. It's not related to PM2.5, and also it's not related to NO2. This is in the smaller sub-study where we have the objectively measured olfaction. So, environment, air pollution, at least at this time, does not explain it. How about the other things? I'm looking at the genetics of Parkinson's, looking at the polygenetic risk score in relation to Parkinson's side by side in relation to olfaction. Not a surprise, very strong association with Parkinson's disease, but not so much with the olfaction, although we still see those dependent association with the with olfaction. So these are the lessons, what we learned from the analysis so far. It's feasible. There are cohorts that has tons of environmental data already collected, and we were able to add in a subpopulation, and we were able to add in a component of Parkinson's data collection into those cohorts without much problem, without the support from the parent cohort. But it's still not easy to study it, because we identify NO2 with PD, but not with olfaction. Why? There are plenty of potential explanations. If we have time, we can discuss about it. But another thing is, so far we only have a snapshot in the data collection for the olfaction, and it will be very interesting to follow them up to prospectively identify olfaction loss as well as Parkinson's diagnosis in the sub-cohort to learn more about this. Now I'm going to switch gear and talk about another cohort that we have been making effort to build a similar research component into. Some of you may know this cohort, the agricultural health study. It's a very unique cohort conducted in Iowa and North Carolina. the participant are only the farmers, the private applicators of pesticide, as well as their spouse. For the sake of, in our data collection so far, I just present the data from the farmers. And back about 20, 30 years ago, they recruited about 25,000 farmers in these two states and collected very detailed farming and pesticide use data. And Dr. Carly Tanner and Freya Kimmel, they added motor symptoms for starting from the phase 2 of the cohort. And starting from the phase 4 of the cohort of the follow-up survey, I added additional non-motor symptoms, as well as the follow-up 5 data collection, which just concluded like a year ago. This cohort is fairly unique. They have 25 years of pesticide data collection, over 100 specific pesticides. So they have very detailed pesticide data. And also they collected other farming and non-farming exposures that common in this population, animal farming, for example. And they have other environmental and lifestyle risk factors. They have diet and DNAs, albeit only from a portion of the population. And they update the health status and symptoms of study population. And Carly and Freya conducted some PD case adjudication up to the P4, and I'm continuing doing the case confirmation for the P5 cases at this moment. Again, we have a motor symptoms collected over about 20-year period, and the non-motor symptoms started from P4. And I'm going to show you some data. Oh, sorry. In 2018, we got a NIEHS grant to do something similar as we did in the sister study. So what we identified again is from the P4 population subgroups of a cohort of 2,500 farmers. We tested their sense of smell. We conducted motor and non-motor symptom screening, and we are following a portion of this participant and trying to conduct a virtual clinical visit. And I'm going to show you a little bit later. And this is a Zoom-based interview, and including neuropsych testing, motor assessment for UPDRS. So again, just to show you the kind of data collection in this cohort and about the symptoms, we listed the 6 most commonly used prodromal non-motor symptoms of Parkinson's and try to look at their relationship to prevalent Parkinson's disease in this case. As expected, very strong association with poor sense of smell and as well as dream inactive behaviors. But interestingly, the association with the poor sense of smell is stronger among the farmers than their spouses, which primarily women, and the farmers are predominantly men. And then I'm going to show you some results on the analysis of pesticide, olfaction, and dream enacting behaviors, with understanding the olfaction and the dream enacting behaviors are the most important prodromal symptoms of Parkinson's disease. So as far as the exposure assessment is concerned, for the pesticide, there are 2 exposures of interest. One is what we call the high pesticide exposure event. The other is the occupational use of pesticide. So the so-called HPEE, we ask about, the occurrence of unusually high exposures due to accidents or spills. So this supposed not just like routine exposures, it refers to the accident. we also ask after the accident, how quickly you clean yourself using the water and soap. And also, which part of the body got exposed? Is that the face or, the hand, the feet, or the body? And did you smell any of those, inhale any of those pesticides? And we also ask about the most massive event, what kind of pesticide involved in the accident. Then typically, occupational use, recall that we have about, at the baseline, 50 different pesticide, but over time, over 100 different type of pesticide. So these are the data about the HPEE, the accident, and purification. We can see that compared to people without such accident, those with the accident were 50, the odds of reporting a purification is 50% higher. And if you stratify by how quickly they clean it up, if they delay for a long time, you can see higher odds ratio. You probably, it's not that much different between derm exposure only and ingestion or inhalation or the internal exposure, but you can see it's slightly higher for the internal exposure. And then we look at which specific pesticide was involved in the event. I think there are about like 20, 30 different pesticides reported. And we then analyze them individually and see which one is associated with higher odds of reporting purification. Not surprisingly, organochlorines stood out clearly. And this has been linked to Parkinson's and Alzheimer's dementia as well. And also some of the herbicide. Some of these have also been linked to Parkinson's disease previously. And then we look at the occupational pesticide use and purification. And in this analysis, we were able to look at the dose-response relationship because we have intensity-weighted lifetime use, considering the higher the numbers representing the higher exposures. We can see, for the organochlorines, we see pretty much dose-response relationship. And also we see dose-response relationship for a couple of the insecticide and also some, herbicide, as we show in the analysis for the accidental spills. We also take a look at the association with stream inactive behaviors. I'm not going to show you the occupational data because the data are fairly consistent, although not as evident as the analysis for the purification. But the dream inactive behavior, the data are very consistent with the findings on the purification. We can see higher odds ratios for ever exposed, kind of dose-response relationship with delays in cleaning up, and more differential between the internal exposures and external exposures. So what's the next step? As I mentioned earlier that we are doing this sub-cohort data collection, we already collected 2,500 participants data with a BSID, tested olfaction, and a lot of details of the motor and non-motor symptoms. And currently, we are targeting about 1,600 of the farmers, 60 to 79 years of age. And we are trying to do a virtual clinical visit, as I mentioned early on, that in a subpopulation to assess their cognitive status, motor deficit, and get medical information as much as possible, and also to reassess their sense of smell as well as their sense of taste. motor and non-motor symptoms. I'm going to give you a quick look at the data from the stage 1, from the 2,500 participants. We collect the data separately for those who self-reported the poor-sense of smell and those who did not. You can see the dramatic difference between the two groups, and also you can see the percent that reported the symptoms is higher than expected in the population. In our sub-cohort, those who self-reported poor olfaction, 22% truly have poor olfaction. But among the people who did not report a poor olfaction, 25% is not a small proportion. So this is my dream, and not come true yet, but we are trying to do this. I'm trying to follow up, either the sister study subcohort or the agricultural health study subcohort, trying to follow them up, and hopefully we will be able to conduct pilot studies to collect biomarkers and microbiome, and to add on some new studies in order to better understand the prodromal development of Parkinson's disease and the environmental contributions to the process. And finally, I just want to thank my collaborators from NIEHS, allow me to use their cohort, even though I'm no longer officially associated with the Institute. And my collaborators at Michigan State, and from Duke, Penn State, Chicago, NIA, University of Washington, and people, helped me along the way in my research, and as well as my funding agencies. And so thank you for your attention, and I will be more than happy to answer questions.
Everyone in the Zoom audience, if you could put your questions in the chat, that would be great. So far, do you have any hints of any, or you haven't looked at any environmental exposures or protocol features?
We look at the first thing we look at is air pollutant and olfaction, with obvious reasons, but we did not identify much of the association. For the self-reported olfaction and dream enacting behaviors, we identify association with pesticide. Yeah, we are trying to see whether we can confirm these associations with objectively assessed olfaction and more detailed sleep disorder assessment. Not yet.
Okay. No. I think given the weather conditions, I think we can stop right now. Okay.
Yeah.
All right.
Okay. Thank you.
So much for an excellent talk. We stop and report it.
Yeah.
Okay. All right. Thanks, guys.
Oh, thank you.
So you're going to be doing some more analysis.